Thank you for the introduction. Thank you for inviting me and the opportunity. I'd like to set the scene on a differentiated, unique, but still to be demonstrated, like most of us, TCR class 2 restricted. Now, there we go. So I won't labor too much about this. We know where we are. Let me just point out the fact that solid tumors remain a challenge. TCRs have an opportunity for a large set of antigens, and indeed there is more than just the killing, and the CD4s may have an opportunity to have a more comprehensive mechanism of action. Sorry, I might have been better here. We all know that TCRs, ideally, they should be at a certain range of affinity. Too low, too high don't work. That's where we want to go. We go there by design or by gene modification. We have a different approach. We came to it by in vivo selection, post-immunization studies. It's good to be in a far-front field with some competition, but not a lot of competition. It kind of validates, but leaves space. And indeed, there are others that are also looking at a class 2 restricted, just so to set the ground. With that said, I wanted to talk to you about Zelluna. So Zelluna is a Norwegian biopharma focused on TCRs for solid cancer. It's very young. It was established in 2016. It comes out of the Oslo University Hospital. It is based on a series of cancer vaccine trials on cancer-relevant peptides. I'll come back to that. In patients that had a very good response, we've collected the material and kept it in the biobank, giving us the opportunity to go and fish for several TCRs. And I'll go more in detail, but the core of what Zelluna is that biobank and the material we can get. It's the class 2 differentiated approach and the antigens that we're targeting, which are some of them inescapable for solid tumors. If I take you back 10, 15 years ago, in Oslo, they were doing some studies where refractory patients with well-advanced cancer, pancreatic, lung, and others, were immunized with a series of simple peptides. The idea was to get the patient's self-immune response to combat the cancer. A lot of patients did not have benefit, but a few had a significant benefit. And from those patients, we've isolated the T cells, which are still in the biobank. And from those T cells, we've isolated the TCR associated with the antigens used for the immunization. And in fact, this is really the basis of our platform, from the vaccine through selection of the TCR, through adoptive cell therapy, i.e. TCR transduction, clinical data or preclinical data, and going on to the clinic. Our TCRs have a high affinity. They're not engineered. They have been in circulation at least in one patient, and they have the ability to be transfected into CD4 and CD8 cells. And partially by design, but also by selection, we have a series of class 2 TCRs, and I'm sure you're familiar with the class 2. Class 1 is a simple approach. It's the kind of the soldier approach, one cell, one kill, or one cell, multiple kills, but direct action. It's kind of that interaction there for the class 1. The class 2 has got a broader, by activating macrophages, dendritic cells, having a CD8 T cell effect with some antigen spreading. So in the end, by that interaction to dendritic cells, that interaction with tumor cells, they can do a series of indirect and direct effects. And I think this is particularly important because we know the challenges for the solid tumors in terms of being able to have multiple approaches, and actually, in fact, having some of that doing in the proximity of the solid tumor, not necessarily inside the hostile microenvironment, but really mastering the entire immune system. In a way, it's kind of a mechanism of action with multiple arms in a single administration. Now, the antigens that have been used were several mutations for K-Ras, wild-type hTERT, and also TGFβRII mutated. They are relevant because of the distribution in terms of solid cancers, but also because the cells cannot really live without them, so they are inescapable antigens. What we're doing is going through a program of safety and efficacy preclinical testing and documentation of those TCRs, able to take us into the clinic. And just to give you a few snapshots of the data, on our class 1 target against TGFβRII, we have against a cancer cell line, colon cancer cell line, the documented antigen recognition by cytokine production, the documented activity to recognize the antigen and kill cells in vitro, so functionality, cytotoxicity, and also xenographic model with colon cancer cell lines. So documenting that the TCR is specific, it's functional, and ready to be tested in the clinic. For the second target, similar documentation here with patient ascites cells and loaded cells with antigen, documenting a specific proliferation, while at the same time an ability to inhibit proliferation in vivo of cancer cells in terms of a control versus the specific TCR. And then finally, for the RAS, you can see we've got multiple targets recognizing different of the genetic variations in G12, giving us, and 13, giving us the opportunity to really target different patients and actually different mutations. Now, trying to catch up on time. Now, where are we focusing our work? We're focusing primarily in adoptive cell therapy. It's really, as you know, taking the sequence of that TCR that I've just described or isolated and actually transducing it through lentiviral transduction into an adoptive cell therapy. So that's the first thing, that's the focus, that's where we're going to document the proof of concept of those TCRs to recognize and potentially have a function in vivo. But like every company that is in this field, we have a version 2, where we will go for a more off-the-shelf opportunity using namely NK cells, since we have proprietary technology to actually TCR, transfect, and get functionality of NKs directed through that TCR antigen recognition. So able us to do what most of us want to do, which is autologous, and have a version 2 to a more off-the-shelf approach. The portfolio that we have, grouped here by the different antigens, you can see that on the first one, in fact, we are already in the clinic in an academic study where the university is using that TCR, but in an mRNA transfected approach, so which we're going to evolve into a lentiviral, and we have the other 2 targets, and we aim to go into the clinic by 2020. We have not disclosed the targets, but they will be solid tumors, and you can anticipate the ones they will be if you look back at the antigens and the prevalence in the different solid cancers that I mentioned to you before. Critical for the success, you will understand that it is to have a strategy for manufacturing, short, medium, and long term, and we partnered with MaSTherCell in terms of doing a process development, taking the academic process develop it, and being it ready for GMP, clinical trial application, IND submission, and then taking it into the clinic. Where are we? So we have the approval and the first patient in the academic. We have the viral vectors established, and we're going to be manufacturing ready towards the end of 19 to go for our own lentiviral first study around the first half of 20, and then a second target behind that, as well as we're establishing our research facilities and the ability. You mean 6 minutes, right? No, but I will give you some time. And as I was saying, and so that this is our milestones and value drivers over the next 3 years. So just a picture of the team, myself and a few others with the experience in small, large pharma, and also some cell therapy. So clearly, and I'm privileged to have this team and have the fun to work with them. And so what are we, in summary? So we have a unique R&D base with a biobank that provides us a unique source of material. We have an established technology for manufacturing, and I showed you that. We're addressing the unmet medical need that I commented in the beginning, and we have a differentiated approach. And if there's anything else, if there's nothing else that you take, please take this. We differentiate it for two reasons: the source of the TCR, form immunization, and the mechanism of action of a class 2 that gives us a unique opportunity, antigen-based and mechanism of action-based, to address the unmet medical need of solid tumors. And I'll go back. And like all of us, we want to be leading, and we have the ambition to be, and I believe we have the chances to be. So thank you very much, and that's it. And I gave you 3 minutes more.